It's time to start. So let me thank Dr. Kim to join us with the presentation about intravital microscopy. And I would like to give a ball to him. Thank you, Dr. Kim, and please go ahead.
All right. So hello, everyone. So my name is Pilhan Kim from Korea. I'm working at the KAIST and also IVIM Technology as well. So today I'd like to introduce about the imaging technology called intravital microscopy for in vivo live cell imaging. So to give you a simple idea, the intravital microscopy is a technique to get this type of a cellular level video from the living animal. So in this particular example, what you are looking at is lung of a sepsis animal model. In green, you are looking at the blood circulation, and in red, you are looking at the neutrophil inside these blood vessels. And then what you are looking at is that this neutrophil, this red color neutrophil, is making a cluster inside this blood vessel and then disturb the blood circulation in this sepsis mouse model lung. So this is a good example to show that intravital microscopy can provide a very detailed live video of the disease development. In this case, it was sepsis in cellular level. So to give you a bigger background, so intravital microscopy can enable a dynamic 3D imaging of various cellular level dynamics in microscopic scale in 3D. And thereby it can provide not just a cellular level information, but also molecular and functional information as well by utilizing the fluorescence probe as well. So these are the three video showing the cellular level dynamics in different organ and tissue like lung, bone marrow, and the skin. And then you can see a lot of various cellular level dynamics such as cell trafficking, basically cell movement and cell-cell interactions and cell microenvironment interactions inside the living clinical animal model in vivo. So additionally, if you're interested in the drug development, this intravital microscopy can enable direct imaging analysis of the to monitor the drug delivery to the target tissue and cell, and then assessing the drug efficacy, and finally validating the mode of action of the drug in various clinical models of human disease. So in this example, I will explain in more detail later. But in this slide example, what we are looking at is the real-time delivery of this lead-present nanoparticle through the blood vessel to the liver in real time. So anyway, so the IVIM Technology's intravital microscopy has succeeded in very high quality in vivo live cell imaging in various tissue and organ of a mouse model. So as you can see in this slide, it includes like a bone marrow, brain tumor, and of course skin, lung, and hair follicle in the skin, tumor vessel, and then pancreas, spleen, and GI tract, prostate, and various lymph nodes, mammary tissue, placenta of the pregnant mouse, kidney, liver, skeletal muscle, heart, trachea, esophagus, and the retina as well. And then we also have experience in imaging sinus, thyroid gland, and various adipose tissue with lymphatic and microcirculation visual imaging. So we are providing Twophoton and Twophoton system together with the research service as well. So I guess maybe many of you already know the details about this microscopy instrumentation, but I just want to briefly talk about it. So for intravital microscopy, we need optical sectioning of the thick live tissue in live animal. So for that purpose, we can use a confocal microscope or Twophoton microscope. In confocal microscope, we utilize the confocal pinhole in front of the detector to reject the fluorescence signal from out of focal plane. And then thereby we can achieve the high contrast, low background, and high quality in vivo imaging in live tissue. And then this confocal microscopy can be a good solution for high sensitivity, multicolor intravital imaging of in vivo tissue. And this right example is the wide area intravital microscopy imaging result obtained from the lymph node of live anesthetized mouse by using our IVIM CM model. So this is the presentation, the real data we obtained from the inguinal adipose tissue of the live animal model in vivo by using our confocal microscope model IVIM-C. In this particular example, we use this mouse offspring of the adiponectin 3 crossed with MTMC mouse. So basically, in this mouse, the adiponectin expressing cell, which is adipocyte, express green fluorescence, and all other cells express red color TD tomato. So as you can see, the green is adipocyte, and the red is mostly blood vessel, and then some other stromal cells as well. So as you can see, we can get very high quality, very high resolution 3D image from this adipose tissue. And then as you can see in this right video, we can get very high quality 3D rendered data from this GSTAC imaging data. So, and then the other technique we can use for intravital microscopy is two-photon microscopy. So we have three models, M, CM, and MS. And then in two-photon microscopy, compared to the confocal single-photon microscope, the two-photon microscope can excite the fluorescence signal only at the focus, like this. So thereby, we can intrinsically achieve the optical sectioning. And then since two-photon microscopy is using lower energy, longer wavelengths near-infrared femtosecond pulse laser, it enables more deeper tissue imaging in live animal. In addition, this two-photon microscopy is also capable of nonlinear multi-harmonic generation imaging, such as second harmonic generation. And the second harmonic generation is a very good technique for non-label, label-free imaging of the collagen in biological tissue in live animal. So this is the real example obtained from the skeletal muscle of a live mouse model by using our CM model. So in this example, when you are looking at it, the green is actually neuromuscular junction expressing yellow fluorescence protein, and blue is the blood vessel, and red is the sarcomere actually inside the myocyte visualized by second generation signal. So this is the real data. This is wide area two-photon and confocal microscopy imaging obtained by using our CM model. So this is wide area mosaic imaging. And actually, this is the single field of view GSTAC sequence we obtained. So in this example, we are looking at the nerve cell, basically. So basically, what you are looking at here in green is the neuromuscular junction, and blue is the blood vessel labeled by anti-CD31 antibody conjugate, which intravenously injected before the imaging. And then also red, this periodic structure is the sarcomere again. And then this is the maximum intensity projection imaging obtained from the GSTAC sequence. And then as this second generation is a very good technique to image the fibrotic collagen in live animal. So this is another example we obtained from the liver fibrosis animal model in vivo. And then also the steatosis of the liver steatosis. So in the beginning, so this is the real GSTAC image sequence obtained from this liver fibrosis mouse model with steatosis. So this green is actually collagen fiber. So you can see this highly deposited, the accumulation of this collagen deposition inside the liver tissue in green. And at the same time, you can image the lipid droplet together in red color by using another fluorophore SF44. So to briefly summarize, the confocal microscope is a very good technique for efficient multicolor 3D intravital imaging, but its imaging depth is typically limited to 100 to 200 micrometer. In comparison, the two-photon microscopy is a better technology if you are interested in deeper tissue imaging, but it has a little bit limited multicolor capability than confocal microscope. But the good thing of two-photon microscope is it has improved imaging depth into the tissue up to 250 to 1,000 micrometer. It depends on the tissue type and organ types. So we are actually providing the all-in-one intravital microscopy system. It's optimized for intravital imaging of live animal model. And then we provide two imaging mode, confocal and two-photon imaging mode. And then it has the advantage of the ultra-fast imaging speed and tissue motion compensation, which I'll explain a little bit more later. The ultra-fast scanning is achieved by using very high-speed rotating polygonal mirror scanner. And then it allows us for very high, ultra-high speed in vivo imaging. Maximum speed is 100 frames per second with 512 by 512 pixels per frame. So it's very fast system. And actually, it is very important to achieve this very high speed in live tissue because of this tissue motion of the live animal. So this is the real raw data we can see from we can obtain from the liver of the anesthetized mouse. So of course, during the imaging, during the intravital imaging, the mouse is live, but it's anesthetized. But still, it breathes air, and then it also heart is beating. So thereby, there are always this microscopic scale tissue motion is present during the intravital imaging. So we developed this motion artifact compensation technique. So basically what we did is we just recorded this high-speed imaging in real time, and then we can compensate the tissue motion frame by frame as shown in here. So this is the result, and then side by side comparison without motion compensation and with motion compensation. From this raw data, we can, by using our motion compensation technique, we can get this high quality intravital imaging from this raw data. Without motion compensation function, you always end up this blurred, very blurred imaging due to these tissue motions. And this is another example. This is the data we obtained from the lung of the live anesthetized mouse. So again, without motion compensation, you will always end up this blurred imaging. So it's very important to have this motion compensation function if you want to achieve very high quality intravital imaging from live tissue and organ inside the animal model. And then we also have live animal maintenance accessories and temperature systems. So you need this tissue, this heater controller to keep the body temperature to be stable during the imaging, during the imaging of the anesthetized mouse. And then we also need a temperature sensor to monitor the tissue temperature during the imaging as well. Okay, so and then before going into the introduction of all of the many various applications, I'd like to quickly mention about our MS2 system. This is the most compact and affordable intravital two-photon microscopy system in the market at this time. And it has a very small, very compact femtosecond pulse laser module inside its microscope. So everything is integrated in this box. So it's very easy to use. So it provides like cost and space saving features, and also easy installation and high usability. And also maintenance is free. So hands-free maintenance because we are using this compact laser system inside. And the all-in-one package, of course, for intravital imaging. So this is the real image data we obtained from the live animal tissue like adipose tissue, skeletal muscle, lung, and ear skin by using our MS2 two-photon microscope system. So from now on, I like to show the real example of this various tissue and organ imaging by using the intravital microscopy technique. Okay, so this is the example of the skin imaging to observe the dynamic immune cell movement inside the tissue, inside the skin tissue. So in this particular example, we use the IVIM GFP mouse model. So in this mouse, it endogenously expressed the GFP, the green pressure protein, in the neutrophil and macrophage in the skin. So we don't need additional staining, additional labeling to image these neutrophil and macrophages. And then at the same time, we labeled the endothelial cell, basically the blood vessel cell, by intravenously injecting the anti-CD31 antibody conjugated with the far-red fluorophore. So in this data, the red color is actually endothelial cell, the blood vessel cell, basically blood vessel wall. And then green is neutrophil and macrophage expressing this IVIM GFP. And in this particular example, we image the ear skin in this setting. Okay, so under this objective lens, we use this U-shaped bracket, and then we put the ear skin against this transparent cover glass here by using saline water. And using this setting, we can image more than several hours, like more than 6 hours at the same site. So in this example, which published in JCB in 2017, what it did was we inject this KRS protein labeled with far-red fluorophore to the dummies of the ear skin by using microinjector. So this red color is actually the injected protein. And then at 0 hours, 3 hours, 6 hours after this KRS protein injections, you can see that this huge infiltration of neutrophil and macrophage. Basically, it's chemo-attracted by this KRS protein to this injection site, as you can clearly see in this video, from 3 hours to 6 hours. And then, of course, the single imaging session can be like 6 hours or even more. And then another good thing of intravital microscopy is not just during the imaging, not just one imaging, we can repeat it because the mouse can be recovered from the anesthetization after single imaging sessions. So basically, we can do longitudinal, repetitive, intravital imaging of the same animal at the same tissue. So in this example, we did cranial bone marrow imaging in the single mouse from day 1 to day 4 after the bone marrow transplantation of this red color bone marrow cells. So from day 1, day 3, and day 4, again, we're looking at the blood vessel at the same time by using anti-CD31 antibody. And then you can see this red colored bone marrow cell, the transplanted bone marrow cell, is engrafted at day 1 like this. And day 3, until day 3, they maintain their positions. And from day 3 to day 4, it now suddenly started to proliferate like this. If you look at this side, so the cell number, so cell area is dramatically increased, started to dramatically increase. So at this day 4, we image it, we take time-lapse imaging like this. Actually, this is the mosaic imaging by using hundreds of the images, hundreds of the single FOV images. And this is the high magnification time-lapse movie obtained from the one of this site. And you can see that this dynamic movement of the and proliferation of this transplanted bone marrow cell. So you can see the proliferation of this cell like this here and here, and the migration of some cell. So this thing, you just saw the proliferation of the cell. in the bone marrow. And this is the preparation of the mouse model for the liver imagings. So in this example, the mouse is anesthetized, and we use the rectal probe to monitor the body temperature together with the heating pad to keep the mouse temperature to be stable, body temperature to be stable. In this particular example, we use the type 2 GFP mouse model. So in this mouse, all the vascular endocellular cells express GFP. So in this mouse, we don't need to label the blood vessel. And in this setting, but this is the real-time 30 frames per second intravital imaging of the liver of this type 2 GFP mouse. So in green, what you're looking at is a liver sinusoid. And then what we did was we intravenously inject a nanoparticle through the tail vein catheter. So now movie just started, and then we started recording when we did this intravenous injection. So as you can see, like about 7 to 10 seconds later, after the intravenous injection, this intravenously injected nanoparticle arrived in the liver through this blood circulation and started to be attached to the liver sinusoid wall like this. And then also you can see this flickering of this image data. It's because the mouse is breathing air. So every time it breathes air, the focus changes up and down. And then since we are using confocal microscope in this example, so thereby, because it looks like this, it looks like flickering. And then this is also the reason why we need this high-speed, ultra-high-speed imaging, more than 30 frames per second, up to 100 frames per second. And then we also use this intravital microscopy over technique to image the various liver disease models, like liver fibrosis, I explained already, and then also liver disease model as well. So in this example, what you're looking at is the lipid droplet in yellow and the blood vessel at the same time. So this liver was induced by a special diet called MCD. And then it's 2 days, 7 days, 14 days, 21 days after starting this MCD diet. And you can see the increase of the lipid droplet, hepatic lipid droplet in the liver. So this is normal diet mouse model, so healthy liver. So you don't see the much of the lipid droplet. And then at 2 days after this diet, you see the increase of the lipid droplet, the small lipid droplet. And then after time, as time goes on with this MCD diet, the lipid droplet size and number increase significantly like this. And then of course we can image the liver in 3D by utilizing the GSTAC imaging capability of confocal and Twophoton microscope. And this is another example obtained from the small intestine. So in this example, the surgical procedure is a little bit invasive actually, but anyway, the small intestine, part of the small intestine is externalized like this, and then make an incision to expose the luminal side of the small intestine, and then we take the imaging at this setting. And then in this particular example, published in JCI in 2015, we used the PROX1 GFP mouse. In this PROX1 GFP mouse, all the lymphatic endocellular cells express GFP. So what we are looking at here is actually lymphatic vessel inside the villi of the small intestine. And then what you are looking at is the transport of this red-colored fatty acid through this lymphatic vessel. And this is the real-time movie showing the contracting behavior of this lymphatic vessel in the small intestine. And then this observed fatty acid in the small intestinal villi is transported through the lymphatic vessel in the mesentery, as you can see here. So from 0 to 90 minutes, you can see this appearance, the transport of this red color fatty acid through the lymphatic vessel. And then we also have a customized system to look at the retina of the live animal model. And these are the imaging data we obtained from the live animal model retina. So we can see the microglia, or we can see the blood vessel together with the flowing red blood cell inside, like this. And now I would like to introduce some of the imaging window technique, includes dorsal skin fold chamber, cranial imaging window, and abdominal imaging window. Now, all this technique was published in the paper, and then we, kind of optimized this imaging technique with our system to make it easy to use for non-expert people. And this is the video we made and then uploaded in the YouTube actually. So this is the surgical procedure for this dorsal skin fold chamber. So dorsal skin fold chamber is a technique to look at the subcutaneous tissue for a long time, more than 2 to 3 weeks. So as you can see, the dorsal skin is stretched, and then let me see. And then we expose the subcutaneous tissue like this. And after that, what we can do was we inject some cancer cell, the processed cancer cell, and then put the glass slides to see it. And then look at the mouse model like this. So this is the real data we can see. So in this example, inside the dorsal skin fold chamber, we injected this GFP expressing human non-small cell lung carcinoma cell, H460. So this green is actually the GFP expressing tumor cells. And then in this setting, we can image this cancer cell together with the microenvironment in the subcutaneous tissue in vivo. And then since we implanted this imaging window, we can image the same animal repeatedly, and then we can easily find the same location at multiple time points. So this is the real example obtained at day 7, day 10, and day 13 with a 3 days interval. And then you are looking at the proliferation, basically the infiltration and proliferation of this GFP expressing cancer cells. And this red colored blood vessel changes over time with 3 days interval. And in this particular example, what we did was we tested some of the anti-angiogenic agent actually. So from day 7 to day 13, without any treatment, the vascular remodeling is huge. So you can see the huge change, the great change in the vasculature without any treatment from day 7 to day 13. But with some anti-angiogenic treatment, we can suppress, we can reduce this vascular remodeling very efficiently like this. And then also by using this dorsal skin fold chamber, we can monitor the nanoparticle delivery or drug delivery to the cancer cell in vivo for a very long time. So in this particular example, we made the cancer genomic model by using triple negative breast cancer cell line, MDAMB231 GFP cell lines. So all this green is the cancer cells, the breast cancer cell. And then through the blood vessel shown in blue by using CD31 antibody. And then we inject the nanoparticle intravenously. And then let's see. So at 2 hours, 6 hours, 24 hours, we can see how this intravenously injected nanoparticle is delivered through this cancer tumor blood vessel to the cancer cells, like this. And then this is the image taken at time point of 2, 6, and 24 hours after this nanoparticle injection. And actually, we can repeat this imaging for much longer time, like even like 2 weeks, more than 2 weeks. And cranial imaging window is a very popular technique for longitudinal intravital brain imaging. So basically, we remove the small part of the skull and then put transparent glass, and then put this pink color is actually the resin to seal this exposed area. And again, we have a YouTube video of the surgical procedure for this cranial imaging window implantations. So basically, we use the small drill to remove the skull, and then put the glass, and then put the resin, and then use the UV to cure the resins. And in this setting, we can image the brain for a very long time. And then we also provide some customization option with this cranial imaging window to have the optimal imaging angle to the brain tissues. So basically, we can rotate the objective lens to get the optimal angle to the brain for the brain imagings through the cranial imaging window. And in this setting, we can image the same mice for a very long time, repeatedly. So this is from day 1, day 4, day 7, and day 10. And you can see the blood vessel shape is maintained, well maintained, but at the same time, you can see the infiltration of this right-hand GFP neutropin into this area, into the meninges actually. And then we can also monitor the function of the blood vessel as well. So in this case, it's the vascular leakage occurred at day 14 after the induction of the general inflammations. So again, you can see the same blood vessel pattern. So we can be sure this is the same site imaged at 7-day interval from day 7, day 14, and day 21. And then what you can see here is the vascular leakage, the leakage of this dye, the Evans blue, from the blood vessel to the brain tissue parenchyma. So, and then this vascular leakage is maintained. It's maintained until day 21, like this. And then later, it induced the huge inflammatory response with the many infiltration of this right-hand GFP cell at this site. So again, this intravital microscopy with the cranial imaging window is a very good technique, very useful technique to observe the certain part of the brain for a very long time in a repetitive manner. And there's another technique called abdominal organ imaging window. It looks like this. So using this window, we can image all of the abdominal organ like kidney, spleen, or pancreas. And this is the real data we obtained from the pancreas and the spleen. And then this is a pancreatic cancer model by using another red color pancreatic cancer cell line, PANC1. And this is the image obtained from the kidney after the renal capsule implantation of the GFP expressing islet cells, and then together with the vascular remodeling. And then this is the actually orthotopic colon cancer model implanted at the cecum here. So green is actually the cancer cell, the CT26 GFP expressing cancer cell, and red is actually the blood vessel surrounding these tumors. And then I like to go a little bit faster. So this is the lung imaging technique. So for the lung, it continuously moves because of the mouse is continuously breathing. So there's a technique called stabilized lung imaging by using lung imaging chamber. And then as you can see here, through this hole, we can suck out the air inside this chamber, and then stabilize the motion of the lung, and it's in this part of the lung for the imagings. And then also we have a YouTube video to show you how to do this surgical procedure to image the lung. So we need to intubate the mouse and then connect it to the ventilator to keep the mouse breathing with these surgical procedures. And then we use this suction-assisted lung imaging chamber. And in this setting, we can obtain this very high quality cellular level imaging of the lung. So this time-lapse video was obtained from the lung of the sepsis mouse model again. And then since we have a very high-speed imaging system, so we can directly see the flowing cell inside this lung vasculature, lung capillaries. So again, so in this example, we use a type 2 GFP mouse. So all the blood vessel is expressing green GFP. And then we inject red blood cell you can see the flow of this red blood cell inside these long vasculatures. And then by using same window technique, we can even see the heart like this. So heart even with this stabilization with the suctions, we can slow down the motion of the heart and stay in some part of the heart. And then with the motion compensation algorithm, which is fully integrated with our software, we can achieve this stabilized high quality image from the lung, the beating heart. And then we can do it, of course, we can do it in the large area like this through the this stabilized area. So all this work has been performed in my lab at KAIST, and then together with the IVIM Technology system as well. And then to test the feasibility of the intravital imaging, we also provide the intravital imaging service as well. And then this is the one of the example to show the in vivo imaging of the exosome delivery in sepsis mouse model, which has been published in Science Advances last year. Anyway, so in this data, as you can see, 10 minutes after the exosome injection, this red color exosome is attached to the surface of the target cell. And then at 30 minutes after the exosome injection, it's internalized to this cell. So this is the kind of definitive demonstration to show the targeting efficacy of this exosome to this target cell. And it happens very quickly, like only after 30 minutes. So for this kind of imaging test, we provide the consulting, and then we also test the imaging procedure and the process labeling, and then optimize the imaging plan. And then if you want to ask for this imaging service, we can provide the quotation at this moment. And then after that, we perform all the procedure, all the process of the intravital imaging experiment, like creating animal model, do the imaging, and the image processing, analysis, and provide the report. So if you're interested in our microscope product and then also our service, then we can provide you the full catalog on your request. So please, if you are interested, please visit our website at www.ivimtech.com or just send the inquiry to the information at ivimtech.com by email. So thank you very much for your attention.
Thank you very much, Dr. Kim. And I hope that someone may have some additional questions. So please, guys, go ahead. Or in case that there is no additional question, thank you very much. And anyway, we are ready to be supportive and helpful on either our email address at Acela or directly to ivimtech.com. And I'll be happy to help myself to anyone who would come up with some question or request. Thank you very much for now.
Yeah, of course. Yeah.
It was a pleasure to have you here. Bye. Bye for now.
Thank you, Kim. Bye.
Hi.